Haddon. Good morning, ladies and gentlemen. I'm Richard Lathrop from the Department of Computer Science at the University of California, Irvine, and this morning it's my pleasant duty to talk about some computationally enabled advances that may move us toward eventually novel cancer drugs. I'm going to begin. The focus of my talk is going to be on the tumor suppressor protein p53, and so I'll begin the talk by telling you about the biological application we address. What is p53? Why is it important? Why do we care? I'll then turn to the focus of the paper, which is the computational identification of a druggable binding site in p53 cancer mutants that reactivates the function and hopefully will lead to killing cancer cells. In particular, this allowed us to produce the first identification of the prima-1 binding site, which had been previously unknown before this work was done. Prima-1 is a p53 reactivation anti-cancer drug that's currently in human clinical trials in Sweden. I'll then turn briefly to biological validation, but because this is a computational biology conference, I'll go through this very quickly. There's a lot more biological validation contained in the paper if you're interested. And I'll close with our use by, again, computational means for anti-cancer drug discovery using the L1S3 pocket that we found. I'll begin with one reactivation mechanism that we found and that's reported in the paper, and then show you a few preliminary results from subsequent work indicating that the pocket has been remarkably productive and that our computational methods, and the good news for you is computational methods are remarkably effective at this task. So, p53 is in the literature nicknamed the guardian of the genome, or sometimes called the genome's guardian angel. It is the human body's central tumor suppressor protein. It integrates signals of cellular distress from a large number of upstream signaling partners, hypoxia in particular for cancer, DNA damage, many other signals, and controls several hundred downstream genes in its role as a transcription factor, including a number of tumor suppressor proteins, programmed cell death, cellular senescence, cell cycle arrest, a number of others. There's more that I didn't have room to put on the slide. It also translocates from the nucleus and migrates to the mitochondria, where it causes cytochrome C release, killing the cell, and a number of other tumor suppression pathways that I'm sure are not yet discovered. All of these pathways are key to your body's ability to suppress cancer tumor initiation and maintenance. And consequently, all tumors must disable these p53 suppressor pathways if they are going to grow into growing tumors. As a result, p53 is the single most mutated gene in human cancers. Half of all human cancers have p53 mutations, and the others are distributed to other partners along the p53 pathways. In particular, MDM2 and E6 are two direct p53 binding partners. They get knocked out in a small subset of cancers, and then the others are distributed through other partners in the pathway. All of these serve to knock out the tumor suppressor pathways controlled by p53. Now, interestingly enough, if you go to the database, about 3/4 of the p53 mutations are single missense mutations in the p53 core DNA binding domain. And the startling implication of this is that about 30 to 40% of all human cancers have full-length p53 that has been inactivated, is functionally inactive, because of a single amino acid change. And this leads to a long-held goal of anti-cancer therapy, to restore p53 function in functionally inactive p53 cancer mutants by means of a small drug compound, small molecule drug compound. I'm showing you here on the right a cartoon sketch. If active functional p53 acquires through normal cellular division a cancer mutant that inactivates it, and you were able to add a small molecule reactivation compound that reactivated p53, then you would restore the p53 suppressor pathways in tumor cells and shrink or kill the tumor. And because of the large number of human cancers that are disactivated by single missense p53 mutations, if successful, this has the potential eventually to save over 4 million lives per year globally. Of course, it's difficult, and there's a large number of reasons it's difficult. I refer to the paper, but just one take-home key is that this drug discovery program falls outside the normal pharmaceutical drug discovery programs and paradigms, which are mostly aimed at modulating enzyme activity, finding agonists or antagonists for enzymes. And this is a whole nother pharmaceutical paradigm to find a molecule that stabilizes and reactivates a destabilized and inactive protein. Nevertheless, there's a lot of experimental evidence that if you could do it, it would actually work. Here I'm showing you some studies that were published recently with mice that have an inducible p53, so they can turn the p53 in the mice off or on. And so at day -4 before, they put in a cancer xenograft into the mouse, and after 4 days it's growing and ugly. They then turn p53 on, and you can see that it clears the tumor. In fact, excitingly, even turning p53 on for a small amount is sufficient to provide dramatic tumor suppression. We think one hypothesis might be that you reactivate the suppressor tumors and they selectively kill the cancer cells. So this is the hope and the dream, and this is what we would eventually like to be able to do with a small molecule drug in human cancers. So now that you know what we're trying to do and why it's important, I want to turn to the use of advanced computation to identify a druggable binding site on the p53 cancer mutants. And in particular, it allowed us, it is the prima-1 binding site, and I'll show you proof of that. So this begins with a serendipitous discovery, something like when Fleming discovered penicillin, and he was growing bacteria in a Petri dish, and a little mold blew in through the window and started some mold, so he had to discard it. But before he washed the plate, he noticed there was a ring around the mold where the bacteria had been killed. It perked his scientific curiosity, and that led to the discovery of penicillin. Similarly here, Chris Wassman was my postdoc involved in a completely different project doing DNA nanotechnology, trying to make little widgets out of DNA, and was in the wet lab doing some wet lab experiments. And he happened to notice on the desk where one of our p53 wet lab research scientists had left a paper saying that prima-1 bound to an unknown cysteine in p53. So he said, I'm a computer scientist, I can check this out. He downloaded a p53 structure from the PDB and did some docking with prima-1, and lo and behold, it bound very nicely to cysteine 124 and oriented it so that it could bind to the, so that its reactive part could bind to the cysteine. It turns out in his hurry and enthusiasm, he had downloaded a mouse p53, and when we tried it on the human p53, there was no pocket, and prima-1 didn't bind anywhere. And in fact, it turns out all of the pockets are closed in all of the PDB crystal structures of p53 at the time. So we brought in the Amaro lab, who has a great deal of expertise in molecular dynamics, did some simulations. Turns out p53 is marginally stable at physiological temperatures. It's sort of always waving like seaweed in the waves. And about 5 to 8% of the time, this little pocket opens up, exposes C124, and the rest of the time it's occluded and closed. Once we started looking, there was a great deal of other computational support for this pocket. We've been involved for a number of years in the study of p53 cancer rescue mutants, which is where a mutation restores functional activity to p53. And on the left, I'm showing you in green a number of p53 cancer rescue mutations out of our database, and they cluster very nicely right around the cysteine 124. This indicates this is an area where small structural changes are able to restore function to cancer mutant p53. Additionally, FTMAP identified a druggable pocket in our open conformation right around cysteine 124, identified in yellow. And then when we started looking, there's even more evidence. It turns out Lambert et al., the paper that my postdoc had seen, had proposed a common binding mechanism for several known cancer rescue molecules that had been reported in the literature. He proposed that they were all able to have an active functional group, shown here in the shade, that alkylates and covalently binds to some unknown cysteine, and this had been reported before. But as I've mentioned, when we do blind docking to the closed p53 crystal structure, it indicates that none of these compounds bound close to any cysteine. On the other hand, when we did computational docking to our open conformation, you can see that all of them dock very nicely to cysteine 124. In all cases, a docking to the cysteine 124 pocket is the best scoring docking of a blind docking across the whole molecule, and they all present their active group right toward the yellow p53 so that they can easily form a covalent linkage. And then once you get the idea that this is what's going on and you charge out to the literature to look for support, it turns out there's a tremendous amount of clues distributed throughout the literature that sort of hint that this is what's going on. And once you have the framework, it rationalizes all these disparate, previously known experimental results. And the interesting thing is, many of the affected amino acids in these results were rescue mutants in the image I showed you earlier of the cysteine 124 surrounded by cancer rescue mutants. So this identified it computationally, but of course any computational result is only a hypothesis, and it requires biological validation to become anything more than a hypothesis. So I'm going to show you that, but again, in the interest of time, I'm going to go through it very briefly. There's more validation contained in the paper if you're interested, but I'm going to skip on to the computational highlights and to some of our more recent drug discovery work. So we made an interesting series of mutants where for wild type p53 and the cancer mutant R175H is the single most prevalent cancer mutant in all human cancers. For both of those, we mutated the cysteine 124 to an alanine, which is a relatively innocuous mutation. Alanine scanning is well known. Alanine is a little smaller. We also did a series of mutants where we exhaustively replaced cysteine 124 with every possible amino acid and showed that none of them were cancer rescue mutations, and so the alanine replacement is not one of the rescue mutants that I showed you. In fact, cysteine 124 doesn't have any at that site. So this is just a control. On the left, I'm showing you Prima-1, which is, as I mentioned, a compound currently in human clinical trials for p53 reactivation. And on the right in green is DMSO. It's the vehicle, the medium. And on the y-axis is the relative increase in cell numbers, and this is with and without prima 1 in the various conditions. The null is cells that don't have any p53. They're simply a negative control. You can see there's no difference between prima 1 in those, as expected. Wild type already has functional p53, so prima 1 has no effect. And again, there's no difference between between prima 1 and the vehicle. C124A, the mutant alone, is essentially identical to wild type. The cancer mutant by itself has large cell growth with just the medium, and as expected, prima 1 knocks it down dramatically. And the mutant that blocks only the C124 binding site has dramatic cell growth and is essentially the same between prima 1 and DMSO. So this pretty conclusively identified cysteine 124 as the binding site because its abrogation knocks out p53 prima 1 functional activity. So we were pretty encouraged by this, and we turned to some drug discovery efforts guided by computational means. In our first initial foray, we did computational docking and other bioinformatics to a series of compound libraries the National Cancer Institute makes available free to researchers through their defined therapeutics program. One of them turned out to be a p53 reactivation molecule, stictic acid, shown here binding into the same pocket for wild-type p53 and 3 different hotspot cancer mutants. Again, you can see that the reactive group is well positioned to make contact with the cysteine, and it fills the pocket very nicely. So stictic acid was one of 45 compounds that we selected to assay out of several thousand from the NCI site, and it turned out to be an active p53 reactivation compound. Here you see a dose-dependent response between different concentrations of stictic acid on the x-axis and the induction of p21, which is one of p53's downstream binding partners that is a reporter for the cell cycle arrest suppressor pathway. And so this demonstrates in a dose-respondent way that stictic acid is restoring p53 transcriptional activation capability. We also showed using differential scanning fluorimetry that it directly stabilized the p53 cancer mutants. Here, the red is stictic acid, and the blue and green are either boiled prima 1 or its active degradation product, MQ, except for the one point out here with wild type where it crashed, stictic acid stabilized the other compounds, or excuse me, the cancer mutants better than either of those. So we were pretty encouraged. We then did a pilot study of 138 compounds. We began with 1.7 million compounds from the Zinc Leads Now purchasable compound database, chose the top 1% by docking into our pocket, and then filtered them for various pharmacokinetic, absorption, digestion, metabolism, excretion properties to get down to 9,668 compounds, and then chose 46 enones, which are the cysteine alkylators, 46 that also bound well with Surflex and had attractive properties of diversity and various other attractive properties, and then 46 based on their autodock. And just for those of you who are curious, the number 46 comes from half of a 96 well plate, which is 48 minus 1 positive control and 1 negative control leaves you room for 46 compounds. So this is a computational reduction of about 12,600-fold from the original 1.738 million compounds to the 138 we eventually chose. We were thrilled that we got 14 active p53 cancer reactivation compounds in the first 91 we've assayed, and we're working on the others and should have them soon. Here's cell viability. Here's Wu. The vehicle with nothing, prima 1 and stictic acid, which I showed you before, and various compounds. The black is P53 null cells. They have no P53, and so they indicate general cytotoxicity. And we're looking for either the R175H or the G245S cancer mutant to be knocked down. Our cutoff is 50% knockdown. So some of these are very weak actives, but others, for example, this one or this one, are actually doing very well. And I should mention that G245S has not been tested for these four. It's not that they're zero. Here I'm showing you photomicrographs of, again, null, of P53 cancer cell growth. If there's no P53, the cells grow very well, indicating no general toxicity. For these two cancer mutants, some of the compounds show dramatic, visually distinguishable cancer cell killing. Here's a dose-dependent response. This is null cells. You can see there's no effect for different concentrations of this particular drug until cytotoxicity just starts killing the cells. But for the cancer mutant, there's a strong dose-dependent response at levels where normal cells are unaffected. And I'm running out of time, so I'm going to skip ahead. But they also reactivate P53 transcriptional capability. 3 years ago, I gave a highlights track talking about P53 genetic rescue mutations with the hope that eventually we would get to small molecule drugs. And I closed with this slide, which I'll close with today. Just remember, this is a very, very long road with a lot of hard work by a lot of people and a lot of blind ends to a new anti-cancer drug. So I've shown you P53 cancer rescue, why it's important, how we used computation to identify a powerful binding site, biological validation, and subsequent drug discovery. I'd like to acknowledge the many collaborators that would help, and at this point stop and take any questions. Identified by? Okay, good question. The question is two-part. First, how frequently was the pocket open in our molecular dynamics simulations? And second, why is it you could not observe it in crystallographic structures? In our experience, the pocket was open about 5% of the time. And this is consistent with NMR studies, which showed that it was open about 5 to 8% of the time. It's not that you can't find it in PDB structures. As I mentioned, we found it in a mouse structure. But all of the human structures that we examined, it's closed. And I don't actually know whether it's closed because there just aren't enough of it to sample the space enough to find something that's only there 5% of the time, or there are less than 10%, but more than 5%, I forget the exact number, amino acid percent differences between human and crystal, and perhaps they affect the flexibility. I'm just not really sure about that.
Yes, it seems that if the target is only available approximately 5% of the time, can you talk about what kind of special challenges that present in getting the drug design to go in there effectively?
Well, good question. And 5% to us sounds like a small amount, but remember that events at the molecular level are happening in picoseconds. nanoseconds. And so something that's open 5% of the time, and the calculations are actually in the paper, corresponds to approximately 10 to the 8th events per second of opening events. And so at the molecular perspective, it's open a lot. And because you get to trade off the binding time versus the concentration, you can make up for that by just slightly increasing the concentration.
Do you think it's likely for resistance to emerge due to additional mutations to P53?
I think resistance is a ubiquitous phenomenon in everything we do. A previous study, I looked at HIV, drug-resistant mutants. And yes, I think P53 resistance is likely to emerge. But these are especially hopeful because they're expected to work well in synergy with other cancer treatments in combination. Because a lot of cancer treatments, especially for the harder to treat tumors, rely on the existence of active P53 pathways for their effectiveness. For example, radiation causes DNA damage and relies on the P53 pathways to trigger cell death in response to the damage. So the general belief in the community as to how you combat resistance is you give several different drugs in a cocktail such that it's much more difficult for the bug to escape from all of them all at once. Instead of if you only have one drug, a single fortunate mutation can escape. So I really envision that P53 will be given in a cocktail with other existing cancer treatments, provide one more thing that makes it more difficult for drug resistance to emerge, and make the other treatments more effective. Good question, thank you.
I think while the next speaker gets set up, we maybe have one more question over here.
Yeah, so I just wondered whether this fast gating that's happening all the time like this, so on average then like 5 or whether you're having a slow gating where you have 5 to 10% of the time open and then shut, and then
So the question is about the duration of the open event. Is it rapidly opening and closing, or is it open for a long period of time?